Hello, it's your boy, the one and only third partner of the department. So I'm just going to read some announcement from our DCAA. So in the meeting of the UP System Academic Affairs Committee yesterday, it was agreed that there will be no suspension of classes this week. Well, but in the context of the surging COVID cases, the ECQ in NCR area and GCQ in Baguio City and the City, all faculty members are enjoined to be more flexible in the understanding of students. They're concerned to waive attendance check for remote classes and to meet synchronous meeting at this time. So the official memo from the PPAA will follow very soon. But for now, instead of doing our synchronous classes, I will record asynchronously for you to follow. So, without further ado, welcome to the wrap-up session for Lecture 5 and Lecture 6, Population Dynamics and Changes in Population over Time.
Before we start our lecture for Module 6, I'd first like to congratulate each and every one of you for producing a high-quality content for Activity 3 and Activity 4. I always brag about it to my Science 11 team, to our Science 11 team, and to my colleagues here in UP Baguio. So kudos for everyone and in the effort, for all the effort that you put in producing all those videos. I'm really proud of you. The second thing that I want to mention for this wrap-up session is that you guys should already have watched the video lecture on Module 5, Population Dynamics, because this wrap-up session will only focus on Module 6, Population Changes over Time in a Living System. Thirdly, I'd like to remind everyone that winter is coming. Yes, after Module 6 lecture, we already have finished the coverage for our first long exam for Science 11. That would be from Module 1 to Module 6. So I hope you guys are starting to review on all of those modules, on all of those lecture videos, which are the main coverage of our long exam. Now, I'd just like to show you our schedule for April. Again, for this week, we should finish Module 5 and Module 5 and Module 6, right? And for the next week, we will have a review break. And for this week, we'll also have a review break because the tentative exam schedule would be on 24 to 25 because it's a departmental exam. So tentative first LE. Okay? So I hope you're getting ready because in April 24 to 25, that's the tentative date for our first long exam in Science 11. I will not meet you in this week, April 11 to 17, and 19 to 23, so that you have time to review. Okay? So that's all. Let me just close this and go back to our lecture. So this is a pretty interesting portion of Science 11 lecture because it talks about, first of all, population. So what is population anyway? When we say population, and if you have remembered our hierarchical system back then, when we say population, these are what we call a group. This is a group of an interbreeding or potentially interbreeding organism. So keyword there is interbreeding. Because when we say interbreeding, they came from the same species. When we talk about population, we talk about those belonging to the same species. They can interbreed with one another. Okay? But aside from being from the same species, a population also, to define a population, and to count them, you need to also qualify the geographical area that they're in and the time frame that they are in. Okay, so that's the main goal of your population. You need to define your species, the geographical area, and the time. Okay, for you to define or to classify a population, for you to count it if you want to count them. Okay, so that's a population. Keyword, interbreeding, geographical area, and at the same time. So for example, the population of UPB students, so they are the same species, well, they can interbreed with one another, and the geographical area there is your UPB campus. At the same time, you can also add for the year 2021. Okay? So that's how we define population in Science 11. So next, recall from our Module 4 that we have discussed about those things that we call genes. What are genes per se? So genes are the things that are passed from your parent to your offspring. These are discrete heritable units of trait that are passed from your parent to you. Okay? And in line of genes, we also talk about what we call alleles. So alleles are what we call alternative forms of the same genes that are found in your chromosome. So, if your gene is the basic physical and functional unit of heredity, which are made up of your DNA, your alleles naman is the variant form of a gene. So, for example, a certain gene can have a different variety or different forms, which are located at the same position or genetic locus on a chromosome. So, take, for example, this chromosome that we have discussed from our earlier lecture with Ma'am Midge. So this chromosome is what I call the sausage butterfly-like structure. Okay, so this chromosome, in that chromosome, you have your DNA. Your DNA is coiled up inside this chromosome, this butterfly-like structure. And in that chromosome, you can find your certain genes. Say, for example, this gene that I put it here, that red one, that red band right there, is the gene for eye color. Okay? It's the gene for eye color. Okay? So that's your gene for eye color right there inside your chromosome. Your alleles are the alternative form of that gene. Say, for example, that gene for eye coloration right here codes for a black eye color. So it codes for a black eye color. Now, on that same genetic locus, meaning on the same position, so meaning right here, on the same position right here, so it must match the same position, okay? So imagine it's on the same position but on a different chromosome, okay? On that same position, you can have an alternative form of that gene. But instead of coding for black eye color, this one, since it's an alternative form, it's an allele, it codes naman for a blue eye color. Okay? So those alleles are the alternative form of the same gene in the same locus, okay, of that chromosome. Okay? On the same position of the chromosome, on the same locus of your chromosome. So there are different alternative forms of that gene. Some may code for black eye color, some may code for blue eye color, some may for olive, some may code for blue eyes white dragon, and things like that. Anyway, so again, in our lecture 4, in module 4, we have also indulged ourselves with what we call the dominant allele and the recessive allele. When we say the dominant allele, this is fully expressed even if you have just one copy of your gene, of that allelic form. So, say for example, in this case, this black eye color, let's name it, this is the allele B, big letter B, and for the blue, let's name this small letter b, the allelic form for your blue eye color. Okay, so if your black is the dominant, you just need one copy for it to be fully expressed. So just one copy of your dominant allele can mask the recessive allele. So even if you have a B and a B, and a B and a B, heterozygous B, and a homozygous B, these are all expressed as black eye color. But for the case of your recessive allele right here, you need a homozygous recessive, all the small B variant, no, the blue variant of that eye color gene, for it to be fully expressed. Okay, so your dominant allele, specifically in this heterozygous format, in this organism with your small letter and big letter B, the black for and the alleles for black and alleles for blue, you can see that just one copy of the dominant black allele can mask your recessive allele right here. So it seems like your recessive blue allele was not expressed in your heterozygous individual. So that's your dominant and recessive allele. Another example is right here. Okay? Here we have the genotype. Again, we review the genotype and the phenotype. What's the difference between the two? When we say genotype, these are the allelic combination. Here we have at least, if you have two allelic forms of that gene, for example, purple and white, so we have P for purple and P for white naman. Purple flower, and P for white, small p for white, okay? Big P ito ito, big P yan. Medyo pangit lang yung drawing ko. Big P. Okay, big P for purple and P for white. Again, your genotype are the possible allelic combination that is found in your, again, why is this like this? Always in pair, no? Why is it always in pair? Because remember, the one came from your mother, the one came from your father. Because we are a diploid organism. Our chromosomes and also our genes are found in pairs. So, we have this allelic combination right here that is always in two letters. Okay? So, for your homozygous dominant, it will code for a phenotype that is purple flower. Again, for the heterozygous individual naman, it will also code for your purple flower because just one copy of the dominant P allele is enough to express the dominant phenotype, the dominant characteristic. And for your homozygous individual naman, you must have both the allelic combination for the small letter P for white flower, for it to be expressed. Okay? So the difference between your genotype is that your genotype is the combination of alleles that is inside your organism. Sometimes your genotype is not always equal to your phenotype. What do I mean by that? Why do I say it like that? I say that because there is this heterozygous individual. When we say a heterozygous individual, it is an individual. That's why it's hetero, because it has big P and small p, the dominant and the recessive allele. It's homozygous right here and right here, because the allelic forms here and here are the same. The allelic forms here and here are the same. Okay? The difference between your genotype are the allelic combination that is within your organism. Your phenotype is the characteristic that you are seeing. For example, your eye color. You can see your eye color, right? Your hair color, the way your hair is wavy or curly or straight. That's your phenotype. That's the character that you can already see. But your genotype is, I imagine it like it's this invisible instruction within our body, within our cell. Okay, moving on. For those of you who are a biology major, you may have encountered the term gene pool in one of your biology classes. But for Science 11, we will define gene pool as the complete set of genetic information contained within a population. So how will you imagine a gene pool? Just look at this figure right here. A gene pool, from its name itself, is the proverbial swimming pool. So imagine gene pool as your swimming pool right here. And an organism can take part in that gene pool if he or she can donate or share his or her genetic information with other members of that population. So say for example, here in the Philippines, for an organism like you guys, for an individual like you, a Filipino individual, to share your genetic, to be part of the Philippine gene pool, you must be able to share your genes, your alleles, your genetic information with the other members of our population, the Philippine population. And how will you do that? You must have sex with the other member of your population, with other Filipino members. Say for example, the Philippines is an isolated island, no? An isolated island, an isolated nation from the rest of the world. But that's how an individual can become a member of the gene pool, through sexual intercourse, through the process of sex. That's the only way he or she can donate or share his or her genetic information with other members. So imagine gene pool like that. It's like a swimming pool of genetic information. And individual of your population can share their genetic information in that gene pool, in that imaginary gene pool. Okay? Why are we talking about that gene pool? Because sometimes, if your organism fails to have sex or produce an offspring in the certain population, in your population, for example, again, for the case of the Philippines, if you cannot, okay, if you do not engage or get involved with the sexual process, you are then excluded from the gene pool. Say, for example, you die, then your gene, the genes that are inside you gets lost. Or you decided not to get a baby because of your own choice. You do not get to be involved or to be a part or a member of that gene pool, because you are no longer sharing your genetic information with other members of that population. Okay? So why are we talking about population? Because sometimes that gene pool, that pool, is dynamic. It's ever-changing. Up until now, it's ever-changing. Sometimes it's changing, but sometimes it's also preserved. But take a look at this question right here. Do dominant traits always predominate in the general population? That's the question. So say, for example, polydactyly. Polydactyly, the condition where an organism, an individual, can have multiple fingers or digits in his palms or in his or her in her toes, no? Okay? So polydactyly, this condition is, di ba, ito yung sinasabi nating swerte, di ba? Anyway, so polydactyly is an autosomal dominant mutation. So it's an autosomal dominant mutation, meaning you only need one copy. So say, for example, this is for polydactyly. You only need one copy of that allele, of that gene, for it to be expressed. So it can either be like that, YB. It can either be like this or like this. The only way for an organism to be normal or have 10 digits or 5 digits per toes or hands, is to have a homozygous recessive. So those two organisms will express a polydactyly, will have multiple digits in their fingers. But only this organism will not express that character. It's a dominant character. But imagine it, no? Why is that polydactyly is not dominant? No? It's not predominant in our population. If it's a autosomal dominant character, if it's a dominant character, and you only need one copy of that gene for it to be expressed, why is that in our population, in the world, no? The normal digits, number of digits, the 10 digits number of your hand from your hands and your feet is the predominant one. How is that so? If it's a dominant character, why is it that it's not always expressed at the time? Okay? Because in population dynamics, that's not necessarily the case. There are certain factors that affect your frequency or the expression of some traits. Another example that I can give you is the case of your ABO blood typing. So if you take, for example, the case of an Asian population, like the case of the Philippines, majority of us have a blood type O, and only about 2% have a blood type AB. But if you look at the blood type of your American Indian population, the Aboriginal Indian population right there, you will notice a slight difference, that majority of them have a blood type AB and only few have a blood type O. So there's this certain changes that allow for the varying phenotypic characteristic and genotypic frequencies in your population. It doesn't mean that it's an autosomal dominant character, that it's always expressed, that it's always present or always predominating the population. It doesn't always mean that way, because there are certain factors that can influence the frequency of your genotype and phenotype in your population. It's not always the dominant or recessiveness of your certain genes. Remember that, okay? Moving on. We look at the changes in the frequency of genes and genotypes within a population. We look at the population level and see how the frequency of your genes, the different alleles, changes over time. So this is really important in understanding how population evolved and how population and how communities and ecosystem perpetuates and continues over time. No? This study will answer our question earlier of why does polydactyly is not so predominant in our population, even though it's a dominant trait. Next up, we'll discuss natural selection. What do we mean by natural selection? From the name itself, it means nature is selecting. She is selecting specific traits in the environment. So meaning, your environment will favor certain traits among others. For example, your skin color. If you are in near tropics or in the tropics, nature will favor a darker coloration. Why is that? Because the higher the amount of melanin in your skin, the suited you are in the environment. Because remember, your melanin, number one, blocks out the harmful rays of the sun, the UV rays that causes cancer. And number two, it blocks out the harmful rays again, that degrades your folic acids in your blood. Because remember, folic acid in your blood is super important for the development of your sperm and the development of your newborn babies. That's why it is an utmost importance that your organism have higher melanin covering in his or her skin so that the harmful rays of the sun won't degrade folic acid. That is super important. Now, from that, you can see that there is this relative fitness, what we call relative fitness of a character. If you are a black person in the tropic and you are a white person in the tropics, so let's say, for example, the black person has 1 or 100% relative fitness, and your white only has 0.5 relative fitness. Because, again, your melanin in the black person will black out the harmful rays and will make sure that your organism will mature to reproductive maturity and will able to reproduce. For your white person in the tropics with less melanin in his or her blood, the harmful rays of the sun will penetrate her skin, may cause cancer, but more specifically, it can cause the folic acid in his or her bloodstream to degrade, therefore reducing his relative fitness. Because fitness in biology is defined, for us biologists, we define fitness as your organism's capability to reach reproductive maturity. Here, the concept, remember this, the concept for us biologists of fitness is not being so macho, it's not being always tall, it's the organism's ability to survive to reproductive maturity and reproducing. If you are able to reproduce, for us biologists, you are very fit. You are perfectly fit. Whether or not you are fat, tall, shaved, unshaved, have beard or not, you are fit for us as long as you can reproduce. You can reach that reproductive age after puberty and you can reproduce with other people. For us, if you can do that, you are perfectly fit. Now, why is that your white people has low relative fitness in the tropics, okay? In the tropics. Because your white people, if they don't put sunscreen or they don't use umbrella, the light rays can cause abnormalities in their sperm and abnormalities in their babies. That's why there is reduced reproductive value here. So again, 0.1, this is 100%, 0.5, this is just 50%. Again, nature favor certain traits among others. The environment will choose what it wants. Us organism will have to adapt to that changing environment. That's why in the tropics, nature selects the black phenotype than your white phenotype. Okay? It favors the survival of your black phenotype than the white phenotype in the tropics or near the equator. So there's that differential survival, differential rate of fitness among characters. Okay? And we always must remember that organism must reproduce. For the genes to continue in that population, the organism must reproduce. If it does not reach that reproductive age, it is not reproductively successful and the genes would be lost forever. One thing to remember here, especially in biology, it's not the survival of the fittest. It's not that. It's the survival of the fit enough. You don't need to be the fittest. Okay? You don't need to be the tallest, the smartest person. You don't need to be that. You need to be fit enough. Because remember, nature will choose what it wants. Nature is dynamic and ever-changing. Maybe now, the taller person was chosen by nature if you are in basketball, but if you move in other field, nature will change its preferences. It will change its preference to other characters. Maybe now the tall genes are favorable in this context, in this environment, but soon it will change. Nature, especially in the natural setting, nature's preference is ever changing. Now it's a desert, but soon enough it'll be an ocean, or soon enough it'll be a jungle. The fittest now may not be the fittest in the future. Do you understand? So that's that. Now moving on to Darwin's theory of natural selection. So for us to understand what Darwin, a naturalist, was thinking back then and in his theory of evolution, just look at this, no? So the number one thing that he noticed is that organisms, your parents beget the same organism. So if this is human, the parent is human, the organism will be human. Okay? Okay, that's okay? If this is a chimpanzee, this will be a chimpanzee. Because organism will produce the same organism. But there are this chance variation in your species, in between species. There is this variation that is always happening. Because remember, from our meiosis, we have the crossing over, right? In your meiosis, there is the crossing over phase where you create here, this crossing over phase where you create certain recombination, DNA recombination, which creates a variety. Instead of just having a pure blue or a pure red, it creates these recombinant chromosomes right here, where you have a tip that is red and a blue middle and so on and so forth. You create at least here 4 different characters, and your organism can have multiple crossing over site that would create much more recombinant DNA, much more chances of recombining, or much more variations for your sperm and egg cell. And in your meiosis also, aside from recombination, what happens in meiosis is that your paternal and your maternal chromosome, the chromosome from your father, the blue one, and the chromosome from your mother, the orange one, can rearrange independently. No? It can be blue, 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 orange, 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 or it can be blue, orange, blue, orange, blue, orange, blue, orange. It can have a multiple of combinations in here. That's why there are a lot of possible combinations in your sperm and your egg. This explains why you and your sister or your brother don't look the same, because the sperm and the egg cells have a lot of possible combinations in them. That's why us as a species is super varied, because variation is important. We love variation because nature is ever changing, and variation will help us survive that ever changing nature, that ever changing environment. Aside from that, Darwin also thought of that that more offspring are produced in each generation. So there is this exponential law of population growth. So there is this exponential law of population growth. So if this is Adam and Eve, there are only 2 persons here, but after a couple of generations, there will be 1 million and so on and so forth. Anyway, so although your population is exponentially expanding, your environment can only support a certain amount of people, certain amount of population. We call that the logistic equation. So, although your population is ever expanding like this, your environment can only support certain cap of population. That's why there is that plateau. Your population will expand exponentially, but to a certain point, it will stop growing exponentially and will reach that stationary pace right here, a plateau pace, where your growth is the growth rate of your population is not that much, because your environment will create that pressure and will limit the growth of your population after a threshold value. Okay? Number 4 from Darwin's theory is that some individual physical or behavioral traits have higher chance of surviving because, again, of fitness. Okay? Of fitness. Your physical traits and your behavioral traits have their degree of fitness. Okay? That will let you survive to that reproductive age and will allow you to reproduce. All of your traits is a conglomerate of all that fitness value from all of those characters that are coded by your genes. Okay? Do you get that? So, specific physical or behavioral traits have also, again, varying survivability. Okay? This is the case of your black skin color or white skin color. They have varying chance of surviving. Okay? That's why certain character survive to this day and certain characters are lost because nature choose that character to survive. And your population does adapt. But the adaptation that we're talking about here is not an adaptation, it's an abaptation. Because when you say adaptation, it implies that your organism act or knowingly change its physical appearance, its physical characters, its behavior to better suit the situation, to better suit the environment that it's living in. Sometimes that's not the case. Your organism doesn't always adapt. Sometimes they do adaptation, meaning your organism gets to be fitted in that environment as a consequence of the character that it inherits from its parent. Of course, those characters were filtered by natural selection of the previous environment. But you need to look at this in this way because your characters that made some of these organisms survive are already present there in the first place before the stimulus takes place. Okay? So that's the difference between adaptation and adaptation. Next, we need to talk about Johann Gregor Mendel. Si father, no? The father of genetics. Because Gregor Mendel is the one that proposed the alternative form or alleles. You got to be proud how Father Gregor made this discovery. How he theorized that there is this thing that we call alleles. Because back then, there were no microscope that was powerful enough to see the chromosome, to see the DNA, to see the genes inside of you. So it was weird that he was able to predict that there was this unit of heredity that was passed between us. And that these units of heredity, these genes, have these alternative forms that may code for this character, the black character, the white character, the purple and the white character, those different characters. And of course, we also have discussed the homozygousness, the heterozygousness. Again, the homozygous genotype would be these two, homozygous dominant and homozygous recessive. The heterozygous genotype would only be this one, heterozygous dominant. Okay, the big B and small b. Moving on. Simply put, your genotype would be responsible for your phenotype, right? But that's not always the case. There are certain interaction that happens that may change the expression of your genotype to your phenotype. That's why I draw this very convoluted scheme here, because it's not always a 1:1 correspondent. It's not always that your your homozygous dominant black phenotype would produce a black phenotype here. There may be interactions of your proteins within each other. The environment can go in here too. Different genes could again interact that may produce your phenotype that is different from your genotype here. But simply put, yep, your genotype is responsible for your phenotype. But I'd like you to put in the back of your head, remember that there is this interaction that is happening behind the scene that may cause changes. For example, you may have the gene for being tall, but since you didn't eat right nutrients, you didn't eat much calcium, you didn't have a balanced diet, that gene won't be expressed. Or maybe you have that gene for tallness, but certain interaction with other proteins from your other genes inside you, gene-to-gene interaction, may have inhibited the production of, you being tall. Now we move to evolutionary ecology. In evolutionary ecology, one of the major breakthroughs back then was the twin vision of Darwin and Mendel on our world. No? Here we have synthesized the theory of natural selection and the theory of genetics. That's why we have evolutionary ecology here. We synthesize these two studies right here. Natural selection and genetics from our father Mendel, si paring Mendel. In evolutionary ecology, the phenotypic variation among individuals within a population is actually a direct result of the interaction between your genes and your environment. Your traits, your physical characters that you are expressing right now, your hair, your intelligence, your height, is actually a direct result from the interaction of your DNA, your genes, and the environment that you're in. They are in constant play with each other. Now, the only question is, what will win? Is it your genes, nature, or nurture? The way that you are brought to life, the way you are treated, the way you eat, the way you behave. So that's the only question. And one of the famous studies that proved it back then was the twin studies. So twin studies, 9/11, no? Okay, that's a joke. Anyway, your twin studies are these experiments of our scientists back then that I don't know if it violated some human rights, but what they did was they collected a couple of twins and separated them by birth. Specifically, your monozygotic twins and your dizygotic twins. The cool thing about monozygotic twins, your identical twins, is that they have the same DNA set. They have the same DNA. You can play with them and assess the influence of your genes on your environment on them. And of course, your dizygotic twins, your paternal twins that are distantly, but they share some genes, but they are not completely identical with one another. Okay. Now, to do these experiments, what our scientists will do is that they will collect a bunch of twins, of course, to have enough sample for their experiment, and they will separate these at birth. No, one would be thrown to a wealthy family, for example. One would be thrown at the poorer family, and this is the Filipino teleserye. No, but the cool thing about it was if these monozygotic twins that was separated at birth and was and they were completely exposed to different environment. Now, if they end up to have the same characters, even if they were raised differently, now you can rule out the influence of environment, and you will say that it was due to their genes, their DNA. Now, if they were separated at birth, okay, and they were reared differently, they were raised differently, but after some time, they develop different traits, you can say that the environment has more influence than their genes. Do you get that? One concrete example here would be for alcoholism, for example. Say, for example, you have a monozygotic twins. You have an identical twin. You separated them at birth. The first one you expose in a good environment, a nurturing environment with a good parents, supportive family, and the other one you expose it to a harsh environment where he had to fend off for himself. This other one would be exposed to a bad parents, no? A harsh environment for him. Now, if at the end of the experiment they both turned out to be alcoholic, then you will say that it's not the environment, it's their genes that causes them to be alcoholic. Okay? But if at the end of the experiment, these two twins, no, became differently, the first one that was raised on a nurturing environment became a good person, a non-alcoholic person. Not good, no? That would be a relative term, but he became a sober person. He didn't drink alcohol, he didn't become alcoholic. And the second one, because he was raised by a very bad parent, he was raised on a very trying family with complete lack of disregard for his growth, for his development during younger years, he became alcoholic. Then you can say that the environment, the way these two children will raise, will affect how they turn out to be. Okay? It will affect if they can become an alcoholic or not. So that's how twin studies work. That's the crux of it. But there are a little bit of variations of that and more sample size. So yeah, those are your twin studies. And what they found out was that in those twin studies, is that sometimes, like for example, for the case of tallness in our genes, no? The genes for your height is not always predicted by your genes. Although a portion of it is caused by your DNA, by your genes, some of it is also affected by your environment. There is that interplay. So the things that you eat can also affect your height. But of course, if you don't have that genes for being tall, you will not be tall too. So there's always this balance. There's always this interplay between these two factors, your genes and your environment. And this was the very funny coinkydink that happened for these two twins. Anyway, next up for evolutionary ecology is the Hardy-Weinberg equilibrium. So what is the Hardy-Weinberg equilibrium? Essentially, the cool thing about the Hardy-Weinberg equilibrium theory and equation is that it can help us predict the evolutionary forces at play that can change the frequency of genes in your population. So how does the Hardy-Weinberg work? Well, first of all, what is the Hardy-Weinberg equation? So essentially, this is the equation. P squared plus 2pq plus q squared is equal to 1. This is your Hardy-Weinberg equation. Okay? So P squared plus 2pq plus Q squared is equal to 1. So why does it look like that? So let's look here. Okay, so what are the P's and the Q's here? For the Hardy-Weinberg equation, your P is the dominant allele. We'll set P for the dominant allele and Q for the recessive allele. Now, it's P squared, meaning that's the frequency of your, again, we'll use the black and blue eye color, no? This is for black eye color. This is for the blue eye color again. The dominant here, recessive here. Now, what do we mean by 2p and p squared? Sorry, that's your ratio of your homozygous dominant genotype. The black, the the dominant genotype, and the 2pq naman represents your, well, the frequency of your heterozygous genotype. Your still a phenotypically black, but it is the heterozygous genotype. And your Q squared represents naman your homozygous recessive na genotype. Is that okay? Is that okay for us? Now, aside from your P squared plus 2PQ plus Q squared is equal to 1, you also need to remember the second equation of P plus Q is equal to 1. What do we mean by P plus Q is equal to 1? Again, your P is your dominant allele, your Q is your recessive allele. So, sabi dito sa equation na to, your ratio of your P allele and the ratio of your Q allele in a population is always equal to 1. If you have a 0.5 P, if P is equal to 0.5, okay? If P is equal to 0.5, your Q is also equal to 0.5. Okay? But if your P is equal to, sabihin natin, 0.25, your Q will adjust to 0.75. That's just plug and play at this equation right here. Okay? Is that okay? Are we clear with that? So these are the basic two equations that we need to see right here. Now, aside from this two, we also have what we call say here for example, your for this case naman, we have black cats that are dominant to white cats. Let's just again use the B here for black cats and the small b, the big letter B for black cats, the small letter b for the white fur cats. Now, if I have an 800 black cats in my population of 1,000, what are my allele frequency? What are my genotypic frequency? And what is the individual, how many individuals are there per genotype? So you can use the equation earlier to answer that. So remember from the question, we have 800 black cats. We have 800 black cats. To get the number of your white cats, we can simply subtract it to 1,000 because all the total of the population was 1,000. Now, the white cats in that population would be 200 because 800 minus 1,000, 1,000 minus 800 is 200. Okay? Now, we set our P and Q. Since here, your black cats, right? This is your black, right? The black cat. The allele for your white cats naman is the smaller letter B. Now, since this is the allele for the dominant, it will be the P, and this is the allele for the recessive, it will be Q. Okay? Now, with that being done, we'll now move on with the computation. To compute for the ratio of your black cats, you simply divide it by 1,000, since that's the total number of your population. So you'll get 0.8. Here naman, that's 200 over 1,000. You'll get 0.2. Okay? Now, remember our equation p squared plus 2pq plus q squared is equal to 1. From that equation, we have q squared. And remember here, if you're looking at this portion right here, your black cats can either be a heterozygous organism, a big letter B and small letter b, or a homozygous organism. Both of these two genotypes will code for a black cat. Okay? But for your white cat, it will only have this genotype, the smaller letter B. Okay? So, since we know the number, the ratio of your white cat, we can now compute for Q. So how will we compute for Q? From this equation, look at this equation. So it says there, Q squared is equal to 0.2. So we can get Q. We'll just square root. We'll have your Q at 0.45. Rounded off to the second decimal place. Okay. Now that we have the Q, remember the next equation that we have mentioned earlier, P plus Q is equal to 1. Since we have our Q, our P would be P plus your Q, 0.45, is equal to 1. You can get your P now by subtracting it to 1. You have your P as 0.55. Again, to the second decimal place. Okay? So now that we have our P and Q, we are now done. The hardest part is over, what I mean. Okay? So you just need to get to P and Q, because your P value and your Q value, these are your allele frequency. Okay? So that's your allele frequency. Now, to compute for the genotypic frequency, again, look at this equation right here. We have P squared, we have 2pq, and we have Q squared. Okay? So, to compute for P squared, you just need to plug in. So that's 0.55, right? Squared. And that would be 0.30. Rounded off again. Sorry for the handwriting. And for this heterozygous individual, that's 2 times P times your Q, which is 0.55. That would equal to 0.50. Oh my god, poor handwriting. And for the Q naman, we have your Q value, which is 0.45. You square that, you have 0.20. 0.20. Again, to check if we have a right value here, you just add them up together. That's equal to 1. Okay? Of course, we round that to the nearest 2 decimal digit. Thanks. Anyway, moving on. Now we are going to... So what are these values that we have seen here? So these are your genotypic. These are your genotypic frequency. Okay, so these are your genotypic frequencies. So again, these value are your genotypic frequency. Since we have your P squared, that would be for your homozygous Okay, homozygous dominant organism. So with both the large letter B, okay, capital letter B. So this is the genotypic frequency of that organism, the genotypic frequency of the heterozygous individual, one big and one small. Both the homozygous and the recessive is 0.50, and for the homozygous individual, homozygous recessive individual is 0.20. So that's your genotypic frequency. Now we have your allelic frequency and genotypic frequency. Now, how to compute naman for the number of individual per genotype? So, how to compute for the number of individual per genotype? So, simple lang ulit. What we will do is get that genotypic frequency. Okay? So, 0.3 times your population. Since we have 1,000 cats, we have 300 for the BB, homozygous dominant individual. So, for the heterozygous individual naman, we have 0.5 times 1,000. Again, since this is the total population of those cats, we have 500 heterozygous organism individual. And we have naman for the last one, we have 0.2 times, again, 1,000 total population. Will have your 200 small letter, both small letter, the homozygous recessive individual. So, yes, those are your number of individual per genotype, your genotypic frequency, and your allelic frequency. Okay? Okay ba tayo doon? So, what are the phenotypic frequency? If you want to check, pala, if you got the right answer, you just add these two. That's 800. And again, since these two code for the black cat, yeah, right? The black phenotypic cat, that's your black phenotype. And this is 200 for the white phenotype that was given earlier. 800 blacks, 200 white cats. So we have confirmed that it's the correct answer. So this is the genotypic frequency. How about the phenotypic frequency naman? How will we compute for our phenotypic frequency? So for the phenotypic frequency naman, we just simply look at the given here. Right? Because this is the phenotypic frequency. This is the physical trait that was already given. Okay? 0.2 for the white cats, 0.08 for the black cats. Or if you're not satisfied with that, you just add these two right here and right here. So this is the phenotypic frequency. You add these two for the phenotypic frequency of your black phenotype cat, and your white cat is right here. Is that clear? I hope that's clear because that is surely going in your exam. Good luck. But that equation back there won't hold true if any of these assumptions right here is violated. The number one assumption is that mating is random. If mating is not random, like for the case of us, where we select people that are tall or handsome or have a fair skin or have a long legs, so on and so forth, the Weinberg that we talked about earlier won't hold true because our mating is selective. Mating must be random for the Weinberg to work. Next is that no new mutation must occur, meaning your organism must not mutate. If your organism mutate, since it is mutating, the value of your P and Q might change. The value of your dominant and recessive allele might change because there's always this thing that comes up. No? It's always changing. Maybe other characters might come up along the way, but that's that. There must be no mutation. Okay? Next is that there is no gene flow from one population to another. What do we mean by no gene flow? When we say gene flow, okay, so imagine these two populations, these two gene pool. This is population A, and this is population B. If your organism migrate from population A to population B, then there is gene flow. For the Hardy-Weinberg to work, there must be no gene flow, no migration. Okay? Meaning, say for example, this is the Philippines. This is the Philippines. Okay? This is the USA. If there is gene flow, there is migration from Philippines to USA, your Hardy-Weinberg won't work. Or maybe it will also get in this direction, but still that is migration. No? There is still gene flow. For your Hardy-Weinberg to work, there must be no gene flow. Meaning, they are completely isolated to each other. Next is that there is no natural selection occurring. Because again, if your P and Q are preferentially selected by your environment, then maybe your Q might die or your P might die. Right? Remember, if they are not fit, if that character, if that traits is not fit, like for example of your skin color, if this is your black, this is your white, no? This is the black skin color. If natural selection occurs, then it will affect the frequency of your Hardy-Weinberg. Thus, it will not hold again. Okay? Because your black is preferentially selected by your environment and your white is being killed off one by one. I mean, 50% of the time. Okay? Remember that earlier. Next is that the population must be large. We must have a large population for this to work. The larger, the better. If you have only 10 population, the Hardy-Weinberg might not work. So these are the major assumptions of your Hardy-Weinberg law. Any departure from any of this will cause genetic changes or variation. Thus, we imagine the Hardy-Weinberg equation as the null hypothesis. Okay? And any departure from it will cause changes. Next, for your evolutionary ecology, natural selection is differential survival and reproduction among phenotype. This is what we were talking about earlier, that nature selects certain things. Nature differentially selects your phenotypic characters because there is this thing that we call fitness. And that fitness value of a certain trait changes. No? So natural selection, it says here, natural selection can lead to change in the population, but then also serve as a conservative force, impeding your population to change. Because natural selection can either increase and also decrease your diversity. If we look at this thing right here, these are the types of natural selection. We have your directional selection, disruptive selection, and stabilizing selection. So your directional selection is a directional in the sense that there will be a change in the number of individuals because nature, for example, what happened during the Industrial Revolution, where, well, before, the number of your white-peppered and black-peppered moth, these two are more or less the same, but during the advent of Industrial Revolution, all those factories, all the suits, it darkened the place. It darkened all the trees. That's why your white-peppered moth was selected against, since it can be seen in darker backgrounds. That's why there's a directional selection that selected your darker colored moth, increasing in pigmentation. Now for the disruptive selection naman, the intermediate organism was the one that was selected against. This is for the case of the snail, where the middle organism gets to be eaten regularly than your blacker colored snail and your whiter colored snail. That's your disruptive selection. The evolutionary pressure is right here. The selection pressure applies on the intermediate organism. Now for the stabilizing selection naman, for the case of birth, the weight of your babies, this is a stabilizing selection since the extremes are selected against. And the middle weight baby, middle weight champion of the world was selected. Because if you have a larger baby, that would put pressure on the mother. It will stress out the mother naman, di ba? If you have a smaller baby, that would cause the newborn to be slightly malnourished, or if it's small, it has low probability of surviving. Thus, the middle character was chosen by nature. Okay? Now, moving on. In evolutionary ecology, evolution by natural selection also depends on the heritability of traits. Sa mana-mana lang yan, sabi nila. At i-compute natin dyan yung heritability index. So, how do we compute for the heritability index? So, simply put, I don't need you to memorize all of this. What I need you to remember is this portion right here. Okay? The heritability index right here. Where an index that is closer to 1, a closer to 1 index means that genetic factor, your genes are the strong influence. And heritability factor that is closer to 0 means your environment naman ang nangingibabaw, ang nag-strongly influence. Okay? So, that's your 4 major concepts of evolutionary ecology. Now, for the last one, we have change due to chance. So this is the clincher. This is the most important one, change due to chance. Because I will say this to you now, chance in biology, in our life, is super important. These random processes are super important in predicting those changes in genetic frequencies. For example, number 1 is genetic drift. So let's look at genetic drift. So genetic drift is the changes in frequency in genotype due to genetic losses. No? This usually result from when you have smaller population, since your smaller population are vulnerable to loss. Yep. And it involves what we call the effective population size, which is the number of adults that are contributing gametes to the next generation. That's the effective population size. The lower the effective population, the higher the chances of genetic drift. So genetic drift are those chance events that could change the frequency of your genotype. Okay? So for mamaya, later on, we'll try to compute for your effective population size, and you will later on see that the number of breeding females and breeding males would be included in the computation of that. So what is genetic drift? So what is genetic drift? Again, genetic drift are those chance events that may cause changes in the ratio of your organism. What causes genetic drift? Again, was mentioned earlier, number 1 is a smaller population size. That's the number 1 cause of genetic drift. A continuously small population is in dire danger of suffering genetic drift. Your founder effect. Yeah. The founder effect phenomenon naman, of course, that's why it's called a founder effect, because when a small band of people moves away from the general population and found a new community. That's why it's founder. They found a new community. And they usually are in small numbers. It causes, since small population ulit, it causes high chance of genetic drift to occur. This is the case for some of the isolated communities, white communities, diba? If you remember, the founder communities of America, the Amish and Mennonites communities that, from Europe migrated to America and founded small communities. And usually these population are resistant to technological change and also to, what do you call this? To mixing with other people outside their community. Okay? And the next one is the bottleneck effect. Bottleneck effect naman is another example of genetic drift when there is also a population size that is drastically reduced. No? So some of the genes may be lost from that gene pool as a result of that drastic event of reducing that population size, that bottleneck event. Okay, so these are the examples of it. For example, the settlements of Tristan da Cunha. So it's literally in the middle of nowhere. Look at this settlement right here. So the story of Tristan da Cunha in South Atlantic is, well, before it was settled by William Glass's family and some other people, some few peoples. So they found the island, they went there and built the settlement. Okay, so after some time, those first settlers and the family of William Glass lived there from a long years, and then by 1961, and by 1961, during a volcanic eruption, since that island is a volcanic island, during 1961, when the volcano erupted, the 300 inhabitants had to move because their hometown, their settlements, is going to be destroyed. So they moved to England. And what they noticed in that population, in the 300 inhabitants, after 2 years of being stranded, settling in that island, what they noticed is that most of them have this thing called clinodactyly. So clinodactyly is like this, where your pinky, the pinky of your organism, of your individual, is slightly bent. It has some slight deformity. Yeah. So majority of them, it's an autosomal dominant trait too. Majority of them had a deformity on the fifth finger. And a lot of scientists back then was appalled, They were surprised because the clinodactyly in population is rare. In general population, in European population is rare. But for the case of that settlement, it has, most of them, contain that deformity in the fifth finger. So they made a study and they traced back that William Glass himself has clinodactyly. That's why the founding population may affect the number or the frequency of genes, even though this trait is not that common in the natural population, in a normal population, since the founder of that island, that settlement, was William Glass, and he has that condition. Most of his descendants and most of the descendants of that settlement has now a clinodactyly. Okay. Another example would be the case of your Pingelap Atoll in I think it's in Oceania. Yeah, it's in Oceania. It's right here, Pingelap Atoll. near on top of Australia, near Tonga and Fiji, all those islands right there, and Papua New Guinea. Yeah, well, near that. So, so Pingalap Atoll is under your Federated Micronesia, no, states of Micronesia, and your Pingalap Atoll has this extreme number of what we call achromatopsia, or color blindness. But this color blindness is an extreme color blindness. That's why it's termed the island of the colorblind. So here is the story of that island. So this is the island right here. Yep, Pingalap Atoll. It's an atoll. Well, from your SpongeBob, I hope you know what an atoll is. Anyway, so here's the story of that. It's a nice island. I would visit this if I have money. Okay, so the Pingalap Atoll is an extreme case of the presence of achromatopsia or color blindness, because back then in 1975, the atoll experienced a typhoon that killed. No, it's like a bottleneck event that killed majority of its population, at least 90% of its inhabitants, and only look at this, only 20 people survived. Okay, and one of those survivors was this man right here, and he is a carrier of a complete achromatopsia, meaning complete color blindness. It's complete black and white. So imagine it like that. And now, in our population, in a normal population like ours, we have the achromatopsia, the percent achromatopsia is very rare. No, very rare for a normal population. But in Pingalap Atoll, no, almost 2.7 or 4.6% of them have achromatopsia or total color blindness. Because back then, this man survived the typhoon, and he was the man that that founded or that repopulated the island. So yep. So yeah. 1 in 33,000. But for them, it's really high, no? The incidence of achromatopsia. Yes, of course, when talking about genetic drift, you must talk about the Amish and Mennonites communities in America. These people are, there are a lot of documentaries about them, right? They are really resistant to technological change, and they don't like mixing with other communities outside. No? And scientists have made a lot of studies in their community, and they have found out that a lot of abnormalities, a lot of conditions are found in them, like your ataxia, bipolar disorder, cartilage hypoplasia, specifically this one, the Ehlers van Creveld syndrome. Look at that head of the baby and the hands. It has also a, what do you call this, extra digits, no? And yeah, polydactyly, sorry. It's also dwarf-ish, no? And so on and so forth. The maple syrup, look at that. You want your urine to be a maple syrup? Anyway, so those things can happen when your population gets to be isolated for so much time. Because if you have small population, you have small breeding population. Okay? If you have smaller breeding population, most of the time, it's highly likely that your those recessive traits, those traits that are deleterious, those traits that causes diseases, gets to be expressed. Okay? That's why it's also bad to marry your pinsan and your have babies with your ates and kuyas. Unless they're step bros. I mean, because if you think about it, when your genetic material looks the same, there is a low variation in your gene pool, your genes. There is highly likely that the recessive traits that causes diseases meet each other. And again, when recessive meet each other and become homozygous, they will express that trait. Okay? So yeah, those things can happen. Scientists actually are saying that, well, for us humans, scientists are saying that it's highly likely that some of us have inbred with each other, because there are a small population of humans before. But now, if you have the chance, no, ito yung sinasabi ko lagi sa mga estudyante ko. If you want high variation, high hybridity in your gene, marry the other race. Marry an American, for example, or an African. It will mix your DNA. And that's good, no? Mixing your DNA. That will dilute the recessive alleles in the gene pool. Anyway, those are my comments here. So we now go to what we call the here 50 or 500 rule as the magic number in conservation. That's why in conservation of your organism, say for example conservation of your lions, your rhinos, you need to at least have a 50 critical number to prevent excess inbreeding, and you need to have a 500 population to prevent genetic drift to happen. When you have smaller population, there is high chance of inbreeding, and when you have high chance of inbreeding again, that is bad for your population, because too much inbreeding, okay, again, as was mentioned earlier, could cause your diseases to meet each other up. Those recessive traits for the disease will meet each other, will find each other, and will be expressed. Okay? Okay, so genetic drift leads to the reduction in genetic variation at the rate of 1 minus 1 over 2n per generation. So this is the equation for computing, this is population of course, for computing for the effect of genetic drift. So again, if you have higher population here, lesser chance of genetic drift to happen, lesser effect of genetic drift. So again, inbreeding is the mating, no? Mating with your brother, with your sister, with your pinsan, mating among close relatives, no? And again, for our population, I will say now, it is bad to be inbreeding with your pinsan. Einstein have done it, yeah, and yeah, a lot of famous individuals have done it. Most of the royal families are prominently always inbreeding because they don't want their blood to be diluted by the plebeians, the normal people. Anyway, so inbreeding, for the case of your Florida panther here, Felis concolor, they are always mating with most of them, their close relative, no? And especially for your lions, no? Your lions are always practicing what we call harem mating. You're always doing harem mating. Only the strongest male can have sex. That's harem mating. And that reduces, no? Since only one male can father all of the children, the father of children, that can decrease, no? Since he is the only one that contributes to the population, that can decrease the amount of variation in your population. That's why they have found out that in your Florida panther, since they are always in breeding, 90% of their sperm is abnormal. So it's they are having a hard time in conserving that organism, maintaining the number of an organism. So this graph naman is to show you how genetic variation can decrease due to certain things, no? So letter C right here is your letter C is first cousin mating. Your letter B is sibling mating, and your letter A is self-fertilization. Look at that letter A. The drop in genetic diversity, in genetic variation, is super steep. Yeah, well, look at this. Your first mating cousin can you can still survive, no? Mas okay pa siya, di ba? First cousin mating than your sibling mating. Of course, that's a no-no. Okay? So, yeah. And again, we are mentioning variation here because variation is important, right? Genetic variation is important. I'm not just saying this to make fun of them, to make fun of these people. But genetic variation is super important especially in our population. Because variation fuels evolution. And variation will allow us to survive. The more varied our population is, again, pag andyan yung virus na yan, or yung alien na gustong pumutol sa mga ulo na matatangkad, no? We will survive kapag may variation. Okay? And also, I want you to look at this figure right here. This is the rule of thumb, no? 50 individuals are necessary to prevent deleterious effects of inbreeding. 500 and up is required, diba? 50 and 500 rule for successful conservation of that organism. And if you look at the graph right here, this is the number of population, okay? Number of population. If you have, technically, if you look at this, what you need to remember is that the higher the population, the better. If you have smaller population, like for this case, your population is just 20, your genetic variation drops quickly. After just 200 generation, you don't have that much variation in your genes anymore. Look at that, no? That's the case of your achromatopsia, the island of colorblind earlier. So we also have this what we call natal homing or natal pilopatry. These are your exhibited by certain organism naman that has this homing process. Kung saan yung this adult individual of that animal, that species, return to their birthplace to reproduce. This is, for example, your aquatic animals, no? Like your turtle and your salmon, Pacific salmon. They usually come back home to reproduce with the same, with the same species that also came back to that same homing place, so same place. That also affects the amount of genetic variation because they will only have sex with the organism that also went back to that same place, okay, to that spawning ground. And here is that computation for effective population size. So essentially, effective population size is 4 times the actual number of breeding males times the actual number of breeding females all over the sum of the actual number of males and the actual number of breeding females. What do we mean by actual number? We are referring to the actual number because not all are contributing to that gene pool. Not all are breeding. Some, diba? Some may still be underage, still not have gone to the puberty phase. No, that's why they're not part of the equation. So that's the effective population size. Now, for a harem mating, like I was mentioning earlier, for the case of your lions, no, that are prominently harem mating, practicing harem mating, where only one, and for walruses then, where only one male will have to mate with all the females, the strongest male. That would lower the effective population size, because the actual number of breeding males right here is lower, right? It's lower. That's why it also lowers your effective population size if you are harem mating. Okay? Now, if we will apply what we learn here, we must think about our very own tamarao, Bubalus mindorensis. So, our Philippine tamarao from 1900s, no? From the 19s, it has 10,000 population. Then it declined through hunting and influence of us. Then nung 1960s, 100 na lang siya. That's why the presidential decree was made that will do a captive breeding program of 20 captives. Now look at that, no? 20 captives. That's a no. That would reduce the variety of them. But yeah, if you can see here, it's slowly increasing the number of your tamaraos. But again, since in that captive breeding program back then with 20 captives, well, I won't blame them, because it's difficult to get funding for this and to maintain those 20 captives. That would cost a lot of money too. But yeah, look at that. So if you're going to set up a captive breeding program for our tamarao, what number of that species will you get to increase their population, to breed them, to maintain their genetic variability too? Okay, so at least you need 50 or 500. But for the case of species that are slowly declining, that's a hard number to reach, that 20. Mahirap na yan. Again, here is a good summary for you. Diba? If you remember earlier, if you have the gene pool, we have the proverbial swimming pool right here. If you have a gene pool, your mutation will plus to the number of genes, will plus to the variety of genes that is there. Of course, I don't know about the US, where they're so afraid of immigrants, but immigrants also increase the number of genes and varieties of genes in your gene pool. Okay? Of course, your emigrants, no, will remove some gene from that gene pool. Yes. And your organism in here will sexually reproduce with one another to create all of those gene combinations. Of course, your environmental factors here would determine what genes would survive and what traits, genotypes, and phenotypes would survive, no? And would have a successful survival in your population. And then if that individual survives, and how well that individual reproduce, and which individuals are chosen depends on nature and the process of natural selection. And that will cause the change in your gene pool. All in all, this is how you should imagine evolutionary ecology, and how, for example, our ABO blood typing changes from a gene pool right here, initial gene pool, to an altered gene pool. This is the reason why for us Asians, for the Asian population, you have the predominating O blood type, but for the American Indian population, you have the predominating AB blood type. And the Mennonites too, the Pingalap atolls, the Tamaraws, all of these are using the same format right here. I don't know, I'm always using blood type because I'm AB blood type. The Dugumbughaw blood type ko no. Yeah, if you go to Japan, for example, well, not all of them, but most of them still are blood races. If you are O blood type, you might get a hard time finding a job. If you are AB, well, they might find you creative. But yeah, don't go to Japan if you're an O blood type. Just joking. So that's it for Module 6. Thank you all for listening. This is Sir Patrick. Peace out.